Many times in this lecture series, I've drawn an epithelial cell. Now I'm going to draw one from the point of view of its membranes, its internal membranes in particular, starting with the nuclear envelope. Now, the nuclear envelope is a double membrane. The inner membrane is associated with a cytoskeleton, the nuclear lamins. We're going to ignore that now. The outer membrane is really continuous with the endoplasmic reticulum. The endoplasmic reticulum is an extensive series of sheets, tubules, and vesicles which pervades most eukaryotic cells and may take up anywhere from 1/4 to 50% of the volume of the cell. And part of the endoplasmic reticulum, the so-called rough endoplasmic reticulum, which includes the outer nuclear membrane, is studded with ribosomes as they translate proteins destined for the secretory pathway. I'm not going to draw those all in. Much of the endoplasmic reticulum might be rough ER in standard cells. Some of the endoplasmic reticulum is smooth ER. The smooth ER has other functions, including lipid biosynthesis or calcium storage, as we've mentioned a couple times. I'm not going to continue drawing those in. The vesicles produced by the endoplasmic reticulum are in transit primarily to the next membrane system here. That would be the Golgi apparatus. Let's not forget that in this cell, there's another important membrane-bound organelle, the mitochondria. Mitochondria have two membranes. The inner one is often highly folded into so-called cristae. The mitochondria are often intimately associated with endoplasmic reticulum tubules, but they are not contiguous with them, although they do get lipids from the endoplasmic reticulum. They do not ever have continuity with the endoplasmic reticulum as far as I know. Back to the Golgi apparatus, there are also a great many vesicles associated with the Golgi apparatus. Indeed, it's almost as if, especially on the outer stack, the trans part of the Golgi, as if it's bubbling away into little vesicles, which it is, in a sense. Many of these vesicles are bound for the cell membrane. There's the ciliary membrane with a special little dip there that I'm going to diagram in blue. Now that is an endocytic vesicle, whereas that is an exocytic one that's getting secreted to the outside. Now the Golgi apparatus here is the main source of vesicles destined for secretion. Those might be directed apically, and they might be constitutive. That is, they might just, as soon as they're ready, go out, or they might be regulated in their exocytosis. They might dock somewhere waiting for a signal. For example, perhaps this set down here has been transported to the basal end of this cell, awaiting a signal to secrete their contents in that direction. Some Golgi vesicles are also bound for internal membrane compartments, endosomes. Endocytic vesicles join endosomes, and as endosomes mature, they become lysosomes, the little bags of death water. Let's put some labels on here before we go any further. Okay, am I missing anything here? Oh, yes. Here is, this is not actually a typical function of an epithelial cell, but this is a site where some bacterium is being phagocytosed, or phagocytosed, if you prefer to say it that way, in which the membrane extends to crawl around some sort of particle being engulfed. Again, that may not be a typical part of the behavior of an epithelial cell. However, I include it because it is a distinct operation of the membrane compared to exocytosis and endocytosis. Here we have this collar of microvilli. Let's say we have a cross section here, and the ciliary membrane. Oops, excuse me. So the general flux here is that biosynthesis of, say, proteins destined for the outside world begins in the endoplasmic reticulum, transitions via vesicular traffic to the Golgi apparatus. Things destined for the outside world travel through the Golgi apparatus. Some of those are destined for internal membrane compartments like the endosomes and lysosomes. Some are destined to go back to the ER, for example, for recycling. Secretory vesicles are destined for the plasma membrane. On the reverse trip, endocytic vesicles join larger endosomes, which may become topologically complex. And then endosomes, although some of the endosomal vesicles also recycle to the Golgi and to the Golgi in particular, they're destined to fuse with lysosomes for degradation. The lysosomes, therefore, are sort of the end of the road here. They're the little bags of death water. Why? Because the internal milieu of a lysosome is very acidic compared to the cytoplasm. In fact, the color code here is drawn along a pH gradient. At some point, you all heard that mitochondria make ATP by exporting protons across the inner membrane and letting them back in to drive the ATP synthase. The internal space in a mitochondrion is somewhere around pH 8. The cytoplasm might be pH 7.2 or so. The Golgi is perhaps pH 6 internally, 6.5 maybe. The lysosomes are down at the other end of the spectrum are pH 4. That's why we call them that. All of this membrane trafficking is really what distinguishes eukaryotes from prokaryotes. The nucleus, yes, that's where they get their name, and that's part of it. But really, all of this extensive vesicle trafficking is something prokaryotes just don't and perhaps can't do. So how do they do it? Well, first of all, why do they do it? We just consider briefly the situation in prokaryotes. A cell like E. coli has a double membrane. There's a chromosome in there somewhere. Big looping thing. And if you want to secrete proteins in a prokaryote, that means ribosomes are right on the DNA, are right on the RNA coming off of the DNA. Let's see, how am I going to color code this here? Here's some RNA, and let's put a few little purple ribosomes sitting on them. They're right up at the membrane, extruding protein products into the extracellular space. So that's the situation in a typical prokaryote. Imagine that this prokaryote learned to deform its membrane somewhat. Now let's skip the outer membrane, which is sort of irrelevant for this line of argument. Imagine a prokaryote that learns to make internal membrane invaginations of some sort. And let's tuck up most of that chromosome in there somehow. And imagine that RNA gets released from the DNA before it's translated, again by ribosomes. Now extruding protein products into a semi-internal space. Let's imagine that they're folding up before they find their way into the outside world in some folded configuration. Well, in the prokaryote, those proteins, as soon as they're secreted, they're basically out there. A very simplified version of the eukaryotic situation is something like this now. The genome is all inside there. Transcripts are exported from this primitive nucleus. Ribosomes engage with them. producing products into an intracellular compartment, which presumably is then secreted with some opportunity for control at each step in this process. There's an opportunity to control whether messenger RNA is exported from the nucleus and engages in translation, an opportunity to control whether these vesicles fuse with the plasma membrane and when they do so, etc. So that's really what a eukaryote gains in part. What a eukaryote gains by all this membrane trafficking. So key to the evolution of eukaryotes, more than tucking the genome away into the nucleus, is the acquisition of efficient ways to bud off little bits of membrane, whether the plasma membrane itself, in this case, or any of these internal compartments, and to address them within the cell. Some protein entering the secretory pathway must have some signal associated with it that says whether it's destined for the outside world or another internal compartment or what. The first part of that is to be able to make membrane vesicles out of some membrane sheet. I'm going to discuss one of the coat proteins that helps do that. Clathrin, there are several others, but clathrin is one of the best understood. Clathrin is a trimeric protein that forms a sort of a triskelion, this three-armed sort of figure. And in association with membranes, clathrin assembles into a cage. That's what the clath part means. So imagine these triskelions lining up together into a sort of a chicken wire. That's enough of that. Surely you get the idea. Now, in association with a membrane, if it were perfectly flat, all you would get is a skeleton to the membrane. But this clathrin cage has a curvature to it, which means that as it develops on a patch of membrane, it induces it to fold up. So imagine this chicken wire sort of cage. My artistry isn't good enough to draw it as it would really look, but imagine this cage progressively assembling on a patch of membrane, and as it assembles, it's pulling it into a bud. Clathrin will dome it up just fine, but it won't pinch it off. That's a role for another protein, dynamin, which is a large GTPase that polymerizes to form coils. I don't really know how big it is relative to the coils, but I'm going to draw this coil with a tight screw thread out of little dashes to represent the monomers of dynamin. GTP hydrolysis by dynamin changes the dimensions of the coil like so. It makes what was this compact but large diameter thread into a narrower coil. You can probably see where this is going. So imagine that clathrin gets as far as making a bud out of the plasma membrane. Dynamin recruits to and assembles a coil around the neck of the bud. And when GTP hydrolysis takes place, it's going to tighten. I'm sure it sounds just like that in there all the time. Narrowing these tubules sufficiently so that membrane-membrane fusion releases a vesicle. So there are several several vesicle-forming coats, excuse me. If we just take a little bit of this and blow it up, let's say here's a bit of endoplasmic reticulum. Shouldn't close it off like that because, of course, the endoplasmic reticulum sort of goes on forever from the point of view of a protein. The immediate destination for most of the vesicles budded off from the endoplasmic reticulum is the Golgi apparatus. I'll just give that 3 layers here, although in reality there are probably several more. That's going to be an endosome. This, of course, is the plasma membrane. This is what we call the trans-Golgi medial because it's in the middle, and cis-Golgi, whereas this is the ER. So these little threefold things, those are going to be the clathrons. Clathrin is involved in endocytosis from the plasma membrane. So I'll just draw some of these in like that. And it's also associated with transport from the Golgi to endosomes. Vesicles that bud off of the Golgi apparatus bound for endosomes use the clathrin coat as well. Let's just make some other icon for the next coat, the so-called COP1 type coats. Conduct most of the transport within the Golgi apparatus. So traffic between Golgi stacks might involve coating by COP1. Note, some of these are going to be going forward, that is, from cis to medial or medial to trans. Some of these events are going to be taking membrane backward through the Golgi stack. Let's see, how about green? Nope, I already used green. I guess it'll be purple for COP2. Now, while I'm drawing, why am I not drawing any coats on the vesicles themselves? Because as soon as vesicles pop off, those coats disassemble. Those coats are about getting the vesicle pinched off, not about targeting it. At any rate, there are these characteristic coat proteins, and there's another one, I don't know how to diagram it really. The retromer mediates traffic out of endosomes. And there's evidence that there are other kinds of coats in there as well, somewhere. So coat proteins and this tubule scission protein, dynamin, they give us a mechanism for turning this whole array of sheets and tubules into a bunch of bubbles. But how do we know which patch is going to bubble out? Once it does, how does it get where it's going? And once it gets there, how do membranes fuse with each other? These are the other problems that the endomembrane system has to solve. Okay, so I'm going to need this space. I should have been writing these labels on the other side, so bear with me a moment. Okay, there we go. So let's see. Imagine some part of the endoplasmic reticulum and some kind of thing that's been manufactured is ready to go. First step is to recruit a coat, and that's the job of a set of coat recruitment GTPases. There are a couple of them. SAR1 is one, and there's an ARF family of small GTPases. That requires that some membrane protein in the endoplasmic reticulum recognize that cargo is ready to go. This thing must be a guanine exchange factor for some sort of cytoplasmic pool of the SAR1 or the RF GTPases. So it must recruit GDP-bound GTPase, stimulate it to exchange GTP for GDP, of course, insert into the membrane as an active GTPase should. This active GTPase recruits the coat proteins, which, if this is the ER, is going to be COP2, which we'll just make it a big sort of roofing tile like that. Okay, so we'll skip the little part with the coat assembly and all, and just proceed. Let's say this GTPase has sufficiently stimulated assembly of a complete coat. Here's a couple of these GTPases in the budded vesicle membrane. Now, once this vesicle pops off with a complete coat, the job of the coat proteins is done. And the GTPase hydrolyzes its GTP, something about membrane curvature or that perhaps the configuration of the coat stimulates GTP hydrolysis. The coat proteins come off. To be reused. Of course, the GTPase, SAR or ARF, returns to the cytoplasm, etc. And then we've got a naked vesicle, uncoated at any rate, but not naked. How does it know where it's going? And once it gets there, how does it fuse with a target membrane? Well, let's deal with the fusion part first. Because it's crowded in here, I haven't mentioned yet that this vesicle has to be assembled with the means for membrane fusion in the form of some transmembrane proteins that look a little like coils. These are V snares. The V is for vesicle. I don't know what this was originally supposed to be an acronym for, but these things come in pairs. Because on a target membrane, which say would be some part of the Golgi apparatus perhaps, there are T snares that match. T for target. Stop and think about that for a minute. There must be, for every traffic route, even within the Golgi apparatus, there must be a compartment-specific pair of V snares and T snares in order for vesicles to identify and fuse with the right membrane. Okay, so that's what happens. These two get together. I'm just going to draw one of the pair for simplicity. The V snare and the T snare recognize each other. Oops, wrong color. Let's start over here. The vesicle and the target membrane, if they approach each other, mate recognition between the V snare and the T snare initiates entwinement the entwinement of the snares is sufficient to mechanically force the membranes close enough to each other that they will readily undergo fusion. That leaves behind heterodimers of the snares that need to be resolved, that need to be untangled by a special complex whose sole job it is to undo that mated pair. But at any rate, that's vesicle fusion. And if this was supposed to have some cargo in it, then it would have been, at this point, delivered to its destination compartment. So again, this mechanism requires that there be mated pairs on each membrane compartment, but the snares are not all there is to it. Addressing is also a function of a large family of small GTPases, the RABs, of which there are something like 24. As a vesicle forms and buds, of course, we've already mentioned the GTPases, the small GTPases that recruit the coats. They must also have figured out how to recruit a RAB family GTPase as a tag, as an address label. So again, this now is a GEF located in the donor compartment membrane, and it must recruit inactive RAB from a cytoplasmic pool of GDP RAB, which must exchange GTP for GDP, insert by its lipid tail, and get itself into the forming vesicle along with whatever the appropriate V snare is, right? So that's got to come along too. Making a vesicle has to include, therefore, not only those coding GTPases, but also these addressing GTPases and the appropriate fusion proteins. So all of that stuff, where, okay, this is, who knows which one it is. I'm just going to say for this hell of it, it's RAB5. Like I said, there's lots of them. We don't even know which compartment we're talking about here. This vesicle sailing off into the cytoplasm with its precious cargo encounters target membranes through proteins embedded in them that are RAB effectors, so called because they're analogous to the effectors that mediate the effects of, say, Rho or RAS or other GTPases. These RAB effectors are looking at approaching vesicles, scanning for the appropriate RAB as an address label. They grab it when they come across it, and the snares can engage and initiate the process of vesicle fusion. Of course, the RAB has to be inactivated at this point and return to the cytoplasmic pool as vesicle fusion proceeds and delivers this cargo to the target compartment. So that's the addressing, that's the vesicle formation part, and that's the vesicle fusion part. Those components all combine to make the postal address system that conducts traffic between the endomembrane compartments within every eukaryotic cell. And it takes many types of coats, dozens of pairs of snares, and dozens of RABs and their associated GEFs and gaps to make it all work.